It's my pleasure to introduce our speaker today. Our keynote presentation by Dr. Jim Winton. He is a senior scientist emeritus at the USGS Western Fisheries Research Center in Seattle, Washington, where he collaborates with a team of scientists working to improve the health of both hatchery and wild fish. Jim is also an affiliate professor in the School of Aquatic and Fisheries Sciences at the University of Washington, and has served as president of the Fish Health Section, on editorial boards of several journals, and as a member of various national and international committees. He's been recognized for this leadership in many ways, and some of his significant awards include the Fish Health Section's SF SNESCO Distinguished Service Award, U.S. Department of Interior Distinguished Service Award, and election to the Washington State Academy of Sciences. And he's authored more than 200 scientific publications across the field of fish health. So I'm very excited for your presentation today, Jim, and I'll turn it over to you.
All right, thanks, Nick. Can you hear me okay?
Yep, sure can. The screen looks great.
All right, good. Well, thank you very much to you and the rest of the organizers. This has been an interesting seminar series. It's really important given the fact that we're not able to meet in person. It's a great way to keep members of the Fish Health Section connected, and it's been a great information sharing. And I also applaud the award winners for the student presentations. They've been universally excellent. So the talk today is a little different than I normally give. It's not a science talk per se. Nick asked if I would talk a little bit towards students and new members of the Fish Health Section, or new members in fish medicine, and talk a little bit about things that might be affecting them as their career advanced. So I've titled my thing Challenges and Opportunities. And so what I've really done is taken some trends that I see and tried to put them together into some sort of themes. So I'm going to talk a little bit about how we got here, a brief history, although if you heard Drew Mitchell's talk earlier, he did a great job on the history of the Fish Health Section. I'm only going to talk briefly about a couple of important points. I've got some trends that I see, and I think others have seen too. These I've grouped into 5 different categories on infrastructure management, physical factors, biological factors, and new technologies. And then I've got a couple of things to summarize. So the first point about how we got here, in the US, fish health activities were largely an effort to keep fish alive in hatcheries, either mitigation hatcheries or conservation hatcheries, or even private sector hatcheries. It was very common for there to be large losses in these hatcheries from disease, and fish health really began as an effort to keep those fish alive. They were led by microbiologists in some agencies, for example, at Leetown at the Eastern Fish Disease Lab, or the Western Fish Disease Lab in Seattle, where I work, or by university scientists. And this was considered by some to be the rise of the bug hunters. These are the people that really started going out to hatcheries, trying to diagnose diseases, assign a causative agent, and recommend some treatments. There were almost no restrictions on fish movements or the use of drugs and chemicals in the early days. Fish moved globally and certainly nationally, and some of the drugs and chemicals that were recommended and used were acutely toxic or carcinogenic and are no longer available, fortunately. 1972, the Fish Health Section was created, and it was the first section of the American Fisheries Society, as you may know. And then some of the fish health centers began to emerge, both agencies and some states, and a few universities began putting together research and diagnostic capabilities to support fish health. Some of the publications from that era, this on the left here, Jim Wood distilled about 30 years of experience into a book on Pacific salmon diseases, which is still an extremely valuable resource, although it's no longer available. The American Fisheries Society published the Fish Health Blue Book, and the Canadians published their Manual of Compliance. And these were the first documentation of methods for the use in fish disease. There were certain state and regional fish disease control policies. The first in the Great Lakes, Jim Warren started the Great Lakes Fish Health Protection Committee, and that has expanded to others in the Pacific Northwest and the Northeast. These regional fish disease control policies involved multiple states and began to try to reduce the fish disease burdens, particularly in wild and hatchery fish in those areas. There were national policies with the updating of Title 50 by the U.S. Fish and Wildlife Service and the Department of Fisheries and Wildlife, or Department of Fisheries and Oceans in Canada. There were then international guidelines began to come into play. The European Union put out guidelines for the member states of the European Union, and the Office of International Epizooties, now the World Organization for Animal Health, was created, and this resulted in the increased regulation of fish movements. Fish now had to be inspected to go interstate or internationally in many cases. There were new restrictions on the use of drugs. This caused the need for veterinarians to be used in fish health centers, and there was more responsibility in the private sector aquaculture for the departments of agriculture. And you can see now the OIE manual. This is the first OIE manual and code. And this is the recent National Aquatic Animal Health Plan by the U.S. Department of Agriculture's AFS. So some of the trends that I think many of us see, and for those of you who have been around the block a few times, a lot of these will be old hat, and I apologize, but for those who are new to the field, maybe some of these things might be useful. These are, in my view, things you'll see during your career. I put first warming due to climate change because this is the 800-pound gorilla in the corner of the room. You will see effects of climate change on fish health, and not only in the private sector, but especially in the public sector and wild fish. And I'll show you some examples. Inputs of toxic chemicals and pharmaceuticals to our waters continue. These have impacts on fish health directly and also on the immune system. The spread of invasive species continues. The habitat loss, changes in the food chain or trophic structure occur, and this can change the prey structure of populations. Expansion of global aquaculture continues in both public and private. Private aquaculture is expanding with many species globally, and public aquaculture is expanding to include conservation aquaculture and enhancement activities. There's an increase in emerging diseases globally, and there's always limited funding for fish health. Another trend is the hatchery wild conflicts out here in the West. That's especially important. We're seeing a lot of discussions about the impact of hatchery fish on wild fish. Increased animal welfare concerns are occurring. There's a movement nationally to begin to, well, actually globally, to begin to limit the consumption and use of animals and also animal welfare that are held by hatcheries. There's a restricted availability of drugs, chemicals, and vaccines occurs. There are still new diseases for which there are no appropriate therapeutants. There's increasingly a need for aquatic animal veterinarians. There's a move towards standardization and accreditation of labs. There are new technologies being developed regularly, and there's a discovery and identification of novel microbes, which produces a new set of problems. Also, a topic that's occurring now with COVID is the evolution of pathogens, and it's important in fish health as well. So I've tried to group those into these kind of categories, and the first is I've listed as infrastructure changes, and I'll try to discuss these as these group topics. So there's limited funding for fish health. This results from competing needs, especially in agencies, but even in the private sector. And sometimes you might wonder if we've been too successful. As I said earlier, it was common 50 years ago for there to be large losses in hatcheries that occurred. Maybe 95 to 100% of the fish would be killed. Those large losses have been controlled to a large extent, and we still have large losses periodically, but they're not the regular event. For example, IHN virus prevented sockeye salmon hatcheries from ever developing until biosecurity technologies were implemented to control IHN exposures. So in many cases, some of the disease impacts have been lessened. There's also been a turnover and a need for new professionals. This loss of institutional memory is important. In many agencies, in many cases, people have retired without being replaced or allowing some overlap to allow mentorship or transfer of information. So this institutional memory about what used to happen at different hatcheries, for example, it used to be common for a hatchery disease person to say, well, we get furunculosis every spring at this hatchery. Well, some of those kinds of information are lost now, and that's important. There's also the need for training and new technologies. As these new technologies come online, we need to know, who gets trained, where do they get trained, how do we do that? Do we hire somebody? Do we send somebody off for training or what? And then there's continues the need for veterinarians to deal with drugs, chemicals, and vaccines. Many of the open positions now in fish health specify the need for a DVM degree to allow this to proceed. There are also laboratory constraints. You need new equipment. It's expensive. Sometimes you don't have the budgets for that. And standardization and good lab practices is occurring. The fish health section has been actively pursuing that, and that's important. There'll be a need in some cases for ISO or other accreditation of laboratories, and this will probably reduce the number of laboratories and allow maybe the standardization of laboratories that are large state-run, say veterinary laboratories of states, universities, to be the main source of diagnostic activities. There'll be management issues. These are, again, competing need for attention and conflicting needs. The genetic habitat restoration, invasive species, et cetera, all are requiring efforts for the agencies, and fish health is often underfunded. There are changing public perceptions, as we said earlier. These hatchery-wild interactions, the roles of genetics and disease have been brought up, and in many cases, these have actually prevented hatcheries from operating. These animal welfare concerns are important and will probably continue to occur and increase. Pollution from hatchery operations has been also a point of contention with the public. We've seen many cases here in the West where hatcheries, net pen aquaculture in particular, has been charged with polluting the environment or releasing pathogens. There's concern about GMOs, such as the Atlantic salmon, use of drugs and chemicals that are in the food chain. And these kinds of concerns are in the public and probably will continue to occur or get worse. Outreach and communications, there's an increasing need for transparency. The idea that you can just do what you want without talking to people is probably over. We need more open access of the agency decisions, or even in the public sector, private sector, aquaculture as well. And this will be important. Also, the need to do something about new infections. For example, when new pathogens are discovered, the first question is, well, is this important and what do I do? And so recommendations have to be made in the face of uncertainty, and the discovery of new pathogens is only increasing. Just for example, in a 10-year period, here are 5 new viruses that have been discovered, and they have all caused huge management concerns. In some cases, large losses, but in other cases, just simply a lot of head scratching and need for additional research on what does this mean. The physical factors that are important include habitat loss. This causes stress to fish, particularly when it's low water, for example, or water temperature changes. So habitat alterations or losses affect species density and disease transmission. The big issue, and I'll talk quite a bit about this, is climate change and global warming. Because of fish are poikilotherms, they respond to temperatures, as you know, and so their immune system, as well as the growth rate of the pathogens, is controlled by temperature. We're seeing global climate change effects in the wild and in hatcheries, particularly in the wild, but the hatcheries are not immune for this. And the inputs of toxic contaminants and pharmaceuticals operate synergistically often with pathogens because they affect the fish immune system. I'll have examples of some of these. So for climate change, as I said, this will change the growth rate of the pathogens at higher temperatures. Bugs, especially bacteria and parasites, grow more rapidly. They can modify the immune response to disease, sometimes make it better, sometimes make it worse. They cause stress that increases susceptibility. They alter the physical habitat. For example, dissolved oxygen levels, especially, and also physical habitat reductions by low flow can cause fish to be held at higher densities in the wild. Can change diets that alter disease resistance to disease, and change distribution and density of susceptible species. So they're really complex changes due to climate change and global warming. And in the wild fish arena, we'll see these being an increasing problem. And this is something that many of you who are working for the agencies will need to deal with. A good example of this is from some work that was done out of John Fryer's lab in the 70s. Still remains one of the only tests that look in detail at the effects of temperature on various salmon pathogens. This one here from Holt's publication, 1975, shows the effect of temperature on columnaris disease. You can see over the top a series of temperatures from 4 to 24 degrees centigrade. These are replicate groups of fish, and the mortality in the controls remained low. But over a very short period of time, from 12 degrees to 18 degrees, the mortality went from nearly 0 to nearly 100%. And this shows the impact of the effective temperature on columnaris. Similar conditions are seen with other pathogens. The sea bream iridovirus again went from 0 to 100% pathogenicity or mortality over a relatively short period. There are a couple of cases where warming temperatures make disease less impactful. Viral hemorrhagic septicemia and IHN virus, for example, the two rhabdoviruses, are more pathogenic at lower temperatures because the fish immune system is not as good and the virus continues to replicate at those temperatures. So it's not all bad news, but it's mostly bad news. So some examples, one of the first really large example out here in the West, and I was asked to use kind of Western examples where possible, was in 2002, where adult Chinook, coho, and steelhead in the Klamath River died in great numbers. This was a large run of fish, had some hatchery supplementation, but in the Klamath that year, there were 50-year low flows and high temperatures from a drought. There were water use conflicts in the upper basin where irrigation water was typically withdrawn, and it was supposed to be put back into the Klamath River to help fish, and the irrigation districts were successful in getting the water turned back onto the irrigators, and the low flows caused these large losses in the lower river. In the lower river, these fish held in pools where these low flows and temperature blocks prevented their movement, and they died from columnaris and ichthyophthirius. This caused a subsequent closure of the salmon fishery. And here are some examples. On the right here, you can see dead fish lining the banks of the Klamath River. This brought tears to the eyes of the Hoopa, Kuruk, and Kuruk tribes that have fisheries in the lower river. Here's a large pool with these dead Chinook salmon in this pool here in the upper left. Columnaris lesions on the gills and ichthyophthirius encysted in the gills cause these fish to suffocate in large numbers. And that was really one of the first wake-up calls here in the West for the impact of climate change, particularly low flows and high temperatures on fish. The impacts of climate change are also going to extend to the winter, where we're expecting higher flows and flashier flows in the winter, which will cause scouring of the redds. And so the impact on these fish is going to be both on the egg stage and on the adult stage. Another example occurred in the Columbia River, which was even larger. This was a large loss on adult sockeye. Adult sockeye had been recovered in the Columbia River and restored from historic low numbers to very high numbers in the 2015 period. We had an unbelievably warm summer out here in the West. We had high temperatures and water temperatures, and this caused a huge mortality in the Columbia River and adult sockeye. Well, and the upriver sockeye destined for Idaho lost more than 90%, and this was perhaps as many as 400,000 fish that died in the river during this period. These low flows and high temperatures early in the run caused this loss, where the fish held in pools in the mid-Columbia dams due to low flow and temperature blocks. The water temperature in the fish ladders was about 25 degrees centigrade, and the fish wouldn't enter them. So they stayed below the lower Columbia dams, where they died of columnaris and fungus. Here's an example, for example, of a fungus-infected fish in the upper right. This was in one of the pools above a dam. Columnaris lesions, again, on the surface and on the gills. On the upper left is the historic temperature trend. This is both at Bonneville and McNary Dam temperatures. You can see they're normally around 16 to 18 during the migration period of these fish. During the 2015 period, the temperature was over 20 degrees in the river. These are pit tagged fish in the lower left. You can see 100% of the pit tagged fish at Bonneville. Normally in the yellow, decline to about 50% up to Idaho. They normally lost some fish during migration, but in the case of 2015, they lost almost 50% of the fish by the second dam. I'm sorry, almost 90% of the fish by the second dam. And so these fish didn't make it. And as a picture in the upper right shows, they suffered these columnaris and fungal lesions and died before they could spawn. There's been even more related climate fish kills. In 2019 in Alaska, this was the warmest temperature in Alaska in history. High mortality in many stocks of returning adult sockeye pink and chum salmon. temperatures in Anchorage were over 100 degrees, and temperatures in at least one river in Alaska were 80 degrees Fahrenheit. Columnaris and saproligni were found with some of the losses, but many of them weren't diagnosed. But many rivers, there were dozens of rivers, had reports of fish mortality, adult mortality occurring in them. Now, some may have been due simply to high temperature, but some were certainly due to disease. Paper by Peter Wesley in Ecology and Evolution documented some of that. And now ongoing right now in out-migrating juvenile Chinook and coho in the Klamath River, again, suffered more than 95% infections from Ceratogena Shasta and 70% or greater mortality due to these high water temperatures and low flows. So the same conditions are occurring in the Klamath, but even earlier this year, the drought in California is severe, as you know. And in this case, the irrigation was shut off, but there was not sufficient flows to flush these fish out of the system. So they've suffered much greater losses to Ceratogena than normal. And this may have, in fact, eradicated an entire year class. And again, the Yurok, Hoopa, and Karuk tribes are really concerned about these kinds of things. They face the loss of their fishery in the future. Also, another important topic is the introduction of pollutants to ecosystems. Effluents from cities and agriculture have a lot of adverse on the health of fish. Inputs of toxins, drugs, antibiotics, hydrocarbons, algae. We're seeing altered temperatures, thermal effluents. Many of these kinds of inputs are increasing the stress on fish or affecting disease resistance. Some of these compounds, like some of the estrogenic compounds from birth control pills, are immunosuppressive and affect the immune function of aquatic animals. These estrogenic compounds alter reproduction and immune function. Some of the metals also are immune suppressive, as are some of the herbicides and pesticides. And some of the systems that have shown impacts are, for example, the Chesapeake Bay, Great Lakes, and Puget Sound. Here in the Puget Sound, Chen et al. Published some work that took advantage and work that Mary Arcouche had done earlier to show that the contaminants in some of the areas of the Puget Sound are affecting the health of fish. This is Puget Sound. The Pacific Ocean is out here to the left, and this is the northern Puget Sound and the southern Puget Sound here. They found that rivers in the southern Puget Sound, the Green and the Squally, had much higher levels of persistent organic pollutants, and also a greater disease burden from nanophytes than fish in the northern rivers. And this suggests that these persistent organic pollutants had perhaps made the fish more susceptible to nanophytes. And so we're seeing these kinds of effects in other cases in the Chesapeake Bay, and others. Luke and others Vicky have shown the impacts of contaminants on the immune function in fish as well. Some of the biological factors that are impacting fish and some of the trends are non-native species. These serve as vectors or reservoirs of pathogens, as well as being invasive on their own right. These trophic changes from industrial fishing and food webs, prey switching, have had changes in both nutritional impacts, which alter disease resistance, as well as having new sources of infection. And then the increase in global aquaculture, both public and private, has been extremely important, as culturing new species in new areas has resulted in the introduction or amplification of pathogens. So we're seeing a lot of pathogens emerge from the expansion of aquaculture, and also from the use of bait fish, which I'll talk about in a minute. So these new and emerging diseases from natural sources, the discovery of new microbes, and the evolution of pathogens then are also biological impacts or trends that you're going to deal with. In non-native species, for example, out here in the West in the Columbia River, American shad were imported to California in 1871, and they've spread all along the coast. In the Columbia River, they've increased numbers to where they actually increase their greater biomass than salmon in the Columbia River, and greater than in numbers as well as biomass. Shad also serve as a reservoir for ichthyophonus, and therefore might be a potential reservoir for disease in Pacific salmon. Another example from the Great Lakes is the round goby, which has now been shown to be an important reservoir of VHS virus in the Great Lakes. So these invasives can be important from a disease standpoint, as well as from simply an invasive species trophic change competition problem. So trophic changes, which are changes in the food web or food supply, can deplete prey species or change food webs. Prey switching can alter the pathways of exposure, as we said, and prey switching can also do aspects of disease resistance tied to nutrition. A good example of the latter are the thymine deficiencies in the Baltic Sea, Great Lakes, and now in California that Esteban talked about at some point. Prey switching can alter exposure to pathogens. We don't have great examples of that, but one potential example is from Ichthyophonus in the middle portion of the Yukon River. From 1999 to 2004, Ichthyophonus continued to increase in the river. Prevalence in adult males and females was greater than 50%. Suddenly in 2005, this prevalence dropped dramatically, and this was correlated to a Pacific decadal oscillation change in the ice cover in the Bering Sea, which changed the biology of pollock in the Bering Sea and may have changed then the exposure of salmon to the pollock that are infected. We don't know that that's the case, but that's one hypothesis to explain the sudden shift in Ichthyophonus in these Chinook salmon. I'm still looking for a better example, so if you have one, I'd like to have it. Emerging diseases are newly discovered diseases never before seen in an area, or known diseases seen for the first time in a new host or a new area. Obviously, COVID-19 is an example of a new host, a host jump. Known diseases with a new presentation or higher virulence are also considered to be emerging. And so we have examples in human medicine right now that we're fighting, but we also have a lot of them in fish. So why are some animal diseases and pathogens emerging? Well, one thing, we're using better assays. So we're looking with better tools. We're also looking at new species or in new areas. So the more we look, the more we find. We're also intensive aquaculture in endemic areas. Sometimes we'll reveal a pathogen that we didn't know was there, but intensive aquaculture will allow it to not only manifest itself, but to be discovered. We're translocating pathogens by aquaculture, by trade, by ballast water, things like that. Also, there's natural movement of vectors, reservoirs, and carriers. Increase in biotic stressors. We're seeing destruction of habitat, changes in food web, introduction of non-natives, and changes in the aquatic environment. So all of these things can facilitate emerging diseases. So the introduction of exotics is one of the most important, and it's often associated with aquaculture or ornamental fish trade. International movements have introduced diseases to many parts of the world. IHN virus has moved from North America to Europe and to Asia. Whirling disease has moved from Europe to the US, etc. So the movements of infected fish or eggs has been important in the spread of pathogens and the emergence of new diseases, or in new areas at least. The use of raw fish or fish products as food has resulted in, for example, bait fish used to feed tuna in Australia caused the largest fish kill ever known. Raw fish also probably introduced IHN virus into aquacultured fish. Recreational fishing, anglers, equipment, bait fish. There's been some great work recently out of both Minnesota and Michigan to look at the effects of bait fish impact. Both Nick and Tom have done great work on some of this and shown how important bait fish are in the introduction of pathogens and spread of pathogens. Other pathways are known: ballast water, migratory fish. So whirling disease, herpes virus in pilchards in Australia, VHS virus in the Great Lakes, ISA in Chile, spring viremia of carp in North America, koi herpes virus globally are all examples of these sort of exotic pathogens that have spread. And this will continue. And if you have a career in fish health that goes for any length of time at all, you'll be involved with suddenly the emergence of some of these kinds of new pathogens or pathogens in new areas. Whirling disease was one example that got a lot of attention because it affected both private and public aquaculture. Introduced to Europe in the 50s, it spread by movement of fish to over half the states. There were declines of up to 90% in certain parts of the Great West, Intermountain West, rivers of Montana, for example. Madison River had 90% decline in its rainbow trout stock. Strains of T-tube effects, the alternate host or the worm host, are important. And we're seeing some resistance in some stocks of rainbow trout, which are helping to recover some populations. And Dr. Jerry Bartholomew and Chris Wilson put together a symposium in 2002 that was excellent in reviewing some of the literature on whirling disease. You're probably very familiar with the Great Lakes. VHS was introduced to the Great Lakes by some method in 2003 or around then. Large outbreaks began to occur in 2005 and beyond. The origin was unknown, but it caused huge losses, as is typical of a pathogen introduced to a stock for a new time, for a new period. So we often see the first exposure of a population suffer from disease introductions, and this was the case in the Great Lakes. These are some photos from Muhammad showing pathogens in gizzard shad, the vector of the VHS virus. Another thing that's important is the amplification of endemic pathogens. This involves endemic pathogens that are in wild fish, but can be increased in aquaculture to high levels. And this is one of the reasons that there are some debates about the hatchery wild fish interactions and some concern about the role of aquaculture and its effect on wild fish, because there is some evidence that these viruses and bacteria can grow to high levels in aquaculture settings that increase the infection pressure on native populations. There's not a great number of examples, but sea lice in some of the net pens in Europe and in North America are thought to be an example. Columnaris, I have one example from Oregon State University, some work that was done there that was a great example. Bacterial kidney disease viruses in marine fish and IPN are examples where net pens or other stocks of fish have been thought to increase the infection in wild fish. The example from Oregon State that's really excellent is in some live box studies that were done with Chinook salmon sentinels exposed above and below a hatchery that had a columnaris and Aeromonas outbreaks. This is the Dexter Pond, Oregon. In 2011, they put a single live cage in the influent of the hatchery and in the effluent of the hatchery. This was for 7 days exposure period, and the mortality was in the effluent was about 25%. And then they took the fish to the laboratory and held them for another 30 days post-exposure, and the fish from the effluent continued to die. So there were significant disease impacts from the effluent, but not from the influent. In 2012, they did the same study with replicate cages. Again, the influent river water proved to be not a risk to these fish, but the effluent did, and for up to 200 feet downstream of the hatchery, they were able to infect live box fish from the effluent of hatcheries. So one of the other things that we've worked on in the lab, Gail Kurath has led a lot of this work in our lab, has been the evolution of virulence and host jumps. And this is intellectually interesting to me, and it's also been kind of in the news with COVID a lot lately. But in wild populations of fish, pathogens are in a different environment. So the fish are at relatively low density compared to aquaculture. There are different species, multiple age classes. They have variation in their innate resistance. They have variation in their levels of immunity from previous exposures. There's intermittent exposures, and the pathogen is generally at a stable fitness peak. That is, hosts and pathogens have generally reached some sort of equilibrium. Not always. If conditions change, that might change as well, but that's typically the case. In hatcheries, however, we have a different situation. We have something of a monoculture with high density rearing, single species and age classes typically. They often have similar genetic backgrounds. There's naive animals introduced regularly. This allows a continuous chain of infection. And so there's no constraint to increases in virulence, and it allows adaptation to a new host. This has been seen a little bit with COVID, for example, where virulence is increased and the adaptation to a new host is continuing with the Delta variants. In fish, however, the best example we have is from IHN virus. This is present in the west coast of North America. Typically, historically, the virus was present from Alaska, well, actually from Russia, Kamchatka Peninsula, but from Russia all the way down to the Northern California coast. And these viruses were genetically at a stable fitness peak and relatively similar over long distances and over many years. There was very little mutation or evolution of the viruses. They all were fairly similar, and we get very similar sequences from fish and stocks of fish and isolates that were quite removed from each other. The virus was introduced to aquaculture in the Hagerman Valley, where it began to evolve into a different strain, adapted more to rainbow trout as opposed to sockeye salmon. And in California, we began to see the virus move into Chinook salmon, again, with its own evolutionary trajectory. This is some work by Shad, all that shows that. This is the Eugenia group here, the upper Gena group, sockeye salmon. You can see these are all short branch lengths. This is a pattern that is fairly typical of an existing long-term infection. So this is a well-established host-pathogen relationship. This is in California. The virus has its own evolutionary tract over here. And the Mgena group from Idaho, again, was different. They all emerged originally probably from the Eugenia group, we think. The Mgena group isolates were moved to Europe fairly early on, where they've taken off on their own trajectory here. And also then the Eugenia group fish were, the sockeye salmon were moved to Japan in the 70s. They began to move into rainbow trout, develop their own evolutionary trajectory, again in rainbow trout in Japan. These were then moved to Korea and to China, where they've continued to evolve. Some of these have been increasing in virulence, and so we're seeing some of the recent isolates from China and some of the recent isolates from Idaho have some of the highest virulence of any IHN isolates that we've ever seen. New technologies are continuing to adapt for identification of microbes. We have environmental DNA, quantitative PCR, digital droplet PCRs, multiplexed assays are all being used now. High throughput sequencing technologies, all these new technologies are being used for identification of microbes. For assessment of immune status, vaccine response, herd immunity, we're developing tools that can look at the antibody response or the cellular immune response. There's an increased need for non-lethal sampling. Both eDNA is being now recommended for non-lethal methods, as are mucus and some other like gills, to be able to take fish non-lethally, which would, especially in private aquaculture, reduce the loss of important or valuable fish. We're seeing new drugs, vaccines, and immune response modifiers being developed. This is exciting because we may have some new technologies to help keep fish alive in some of these cases. Especially hatcheries. There's not so much you can do about wild fish for that, but there are also microbiome and metagenomic analyses. And there's a big question about training needs, as I mentioned earlier, is who gets trained, how do we get that training into the lab, and how do we get the equipment and stuff to perform that training. And then again, novel microbes, the so what question. I showed you some of the new viruses before. We talked about that. As you discover new microbes, there's always the question about what do you have to do about this. So some of these new technologies involve, for example, environmental DNA here on the upper left. These are river water samples being collected for eDNA. Here's a small benchtop quantitative PCR machine. This is a MinION nanopore technology. It's a little portable sequencer that you can take to the field and do DNA or RNA sequencing. And this is the Fluidigm disc capsule cartridge. It's a nanoliter cartridge for multiple quantitative PCRs. You can do up to 48 PCRs on a single chip. These can even be multiplexed, but you can look for 48 different pathogens at the same time in nanoliter volumes. And so some of these technologies are now being employed for fish health, where it increases the throughput, the information you acquire, and also the speed of the analyses. And so as these technologies get used more and more, laboratory workers and pathologists are going to have to be more and more knowledgeable about some of these kinds of tools. So in summary, it's really no longer sufficient to simply identify pathogens or recommend treatment in hatchery fish. Fish health is much more complicated than that. There's going to be a move towards integrated fish health programs, especially in agencies that fish health programs will have to operate within the conflicting needs of management. For example, here in the West, there's a real need to open up habitat for fish. And so they're removing weirs and barriers in allowing fish migration above hatcheries. This has a big fish health impact because now we have spawning fish in your hatchery water supply. And before, the water supply was free of some of these pathogens. And so we're now seeing these programs where there's conflicting needs to management needs that have to be addressed. Future fish health management will be more complicated. It'll have both wild and hatchery fish will have to be considered. as well as both infectious and non-infectious diseases and stressors. And so fish health will now be both infectious and non-infectious. So the things like the thymine deficiencies or other non-infectious diseases like toxins and contaminants will be important to study as well. There'll be increasing attention to fish health by the public. This is not going to go away. And so you'll have to continue to operate in the public sphere and be able to inform the public about what's happening, what's going on. For example, the current fish kills in the Klamath have received a lot of media attention, and it's been really important to address those issues. There's a trend towards specialized diagnostic labs separate from the clinical aspects. And so we'll see laboratories that are now certified or accredited being used for much of the fish health diagnostic work. Other laboratory workers will be doing more of the clinical aspects of looking at the fish, collecting samples, or diagnosing the disease. Recommending treatment for the diseases. One of the things that I'm particularly concerned about is the need to preserve linkage to research. In the past, many of these activities were done either in universities with research activities or in agencies with which there was a clear linkage to research research scientists in laboratories. And as these fish health activities get more specialized, that need to link to research may be lost. And I'm concerned about that because it's important for researchers to know what's important, what's going on, and what they should be working on. So I think that's an area that I'm watching as well. So with that, I'll end this thing and try to take a few questions if we've got time. And again, thank you for your attention and appreciate your help.
All right, thank you, Dr. Winton. That was a very interesting talk. We do have plenty of time for questions if folks would like to unmute themselves and ask Jim directly. Otherwise, you can put it in the chat box and I can help facilitate that. Would you like to stop sharing your screen? I can.
Oh, sorry. Oh, shoot. Yeah, my share bar. Can you grab it? My share bar is gone again.
Okay. I will work on that.
Oh, here it is. Wait a minute. I got it. Okay. Does that work?
There you go.
Okay.
Jim, excellent presentation as always. I think that last point that you parted on is a really interesting one, and regarding that potential, the break in linkage between some of the research and some of like being at the forefront of things. I wonder, do you have any recommendations for folks? What or suggestions on how do we approach that? Because I share your concern on that.
Well, it's a difficult issue because many of these labs, we've worked a lot with the Washington Animal Disease Lab, Waddle at Pullman, and many of them receive samples and they have in the case in the private sector, at least have a kind of a client-patient relationship, a laboratory patient relationship, at least, that doesn't allow them to really talk about stuff very much. And so there's times when we don't really know what's going on. And we've also had some trouble getting samples because not only is it kind of an extra burden on their time, but expense. And so getting isolates or getting things sent back out of these laboratories sometimes is difficult. And so we've tried to work with them and tried to work, for example, with the submitters. In the case of the Fish and Wildlife Service or other agencies, we've had more luck where, for example, agencies can make a requirement that the disease labs will give us back isolates or samples that they've had. That's less common in the private sector, which is often not so interested in having everybody know about what pathogens they have. And so I am concerned that we'll make some progress with the agencies, but maybe not so much with the private sector.
Hi, Jim.
Hey, Marty.
You mentioned some of the new multiplex assays and the deep, I don't know if you got into deep sequencing, but I think I'd appreciate, you alluded it to one of your questions before, is the need for determining what's it take when you just find the difference between finding a microbe and finding the disease. Maybe, so there's got some new members here. Maybe elaborate on the importance of that from your experience.
Yeah.
Maybe you can give an example.
All right, well, thank you. Well, there's plenty of examples. I listed several of them. In some cases, we'll find a pathogen or we'll find an agent, let's say. And there's always an excitement about new things and trying to make more of them sometimes than they really are. And so there's often a tendency, at least, to try to talk about these things as being more important without completing Koch's postulates and doing some laboratory tests or something else. I think it's really a little bit concerning that we need to be careful about the difference between a microbe and a disease agent. So not all microbes, of course, are pathogens. Only a small subset are. And many of these may be just simply commensal, so to speak, in the population, or in some cases, at least, we've isolated microbes that actually induce an immune response and are protective against other real pathogens. And so oftentimes this gap in information is an opening for people to assume that something is worse. In the case of, let's say, Piscine reovirus in the Northwest, something you're intimately familiar with, there's been the lack of information allowed an opening for people to assume things that were probably not true. And so we have to be kind of careful. And this goes again to the need for research on these things. We can't simply allow a discovery of a pathogen to sit there and not work on it. And so in many cases, I think it's important to continue the research to find out, is this really something we need to worry about or not? But until then, we need to be careful about what we say about it and the information we release.
Okay, thanks.
All right, any questions out there?
Hey, Jim, I have a question. It was a really nice description of the sort of the evolution of the fish health field from, the classic bug hunters to the development of therapeutics and prophylactics. So I guess what I would call the modern era of ecological disease. Along with this transition, I guess we've seen a change in the portfolio of our customers. And I was wondering if you could address how you feel that the fish health professionals have adapted to those new customers, and if there's room for improvement in how we might engage more actively with this new era of ecological customers.
Okay, Paul, thanks. That's a really excellent question and a little bit of a difficult one, because as you know from your work, you're now dealing with a different customer base, and so are several other people. And so I think it's going to be incumbent on fish health people to be cognizant of both public information and how we deal with very diverse audiences and how we communicate our findings and our results to them, and also how we structure our research portfolio. In many cases, as you've done, the research has broad implications for very large scale populations in the wild. And so you've got a whole customer base that we didn't used to have when we just talked about hatcheries. And so I think it'll be important as we go forward to consider that much wider base, and both as far as granting agencies, but also as customers, and who we communicate with and what we say about it, because it will be a much more complex environment, but it's also going to be a more exciting and important environment.
Great. I'm curious, Jim, when thinking about training the next generation of fish health folks, the shift that you described towards more specialization, I see that too, but yet the stressors that you described were much broader than a single pathogen or a technique for diagnosing it. Yeah. How do you balance this training for more of like a One Health approach versus a traditional microbiologist on the bench?
Yeah, well, that's difficult, because in the past the specialization was relatively easy. I remember when you could fit all of the viruses known of fish in one small 35 millimeter slide, and all of the bacterial pathogens fit on a second bacterial slide, and the parasites all fit on a third slide. And so that isn't true anymore. And So not only are the numbers of pathogens or range of pathogens different, but the techniques used and the species in which they occur and the settings in which they occur are more complicated. And so your point is really well taken. It's relatively easy to be a specialist and excellent in an area, and we need those people, but we also need those generalists. And so the people that used to be able to know, for example, all of the diseases of all of the species in their agency really are kind of rare now. And so we're not seeing those people with that long historic experience in many cases. And we need to try to rebuild that somehow, because those sorts of broader integrated approaches are going to be important, as well as the specialization about how to diagnose a certain virus or this small group of viruses. But yeah, I share your concerns about that. I think that's an important question.
All right, any other questions out there? There's nothing in the chat box right now, except for some compliments for you, Jim, on an excellent presentation.
Well, thank you. I enjoyed it. It's, as I said, it's a little different than I normally talk I normally give, and so I wanted to cover quite a bit of stuff, and I probably went too fast, but at least it'll be recorded, so maybe we can play it at slower speed or something if you want to hear some of the stuff. But anyway, I enjoyed it. I think it's important to once in a while have a little bit of perspective, and so maybe that helped a little bit.
Definitely.
Okay.
Drew,
Great talk.
question?
Oh, hi, Drew.
There he is.
Yeah. I lost everything. Yeah, I'm good at this. I just want to say great talk, and it's you've been a real standard for this whole industry. Thanks for your input. And I wrote in my comment, I think this is something that should be required viewing for students entering fish health. So again, congratulations.
Probably only if they misbehave, then you have to make them sit in a room. As I said, Drew, nobody knows more about history than you do. Your history keynote was exceptional. I didn't realize the fish health went back to the dark ages, so that's great.
Well, thank you very much. This is a great way to end the summer seminar series. Thank you, Dr. Winton, for your presentation.